A very good morning, all of you. Myself, Dr. Rupali Nanekar, Assistant Professor, Department of Biotechnology from CKT College, welcome you all in today's lecture. Students, today we are going to study about DNA replication. So let's start with today's topic. As you know that replication is one of the important and vital thing in genomes and gene, both in prokaryotes and eukaryotes. Replication of DNA is important for transmission of gene from progeny, from father, mother, or from the parental to your next progeny. It's a generation to generation kind of scenario taking place in eukaryotes as well as prokaryotes. DNA replication of chromosomes, duplication in bacteria and eukaryote requires certain important enzymes and protein for replication. Without that, it is impossible for carrying out any kind of replication. Coming to the DNA replication model, which got a popularity in after 1953. As you know that Watson and Crick proposed a model of double helix where they realized that whenever there is a DNA replication happening, it needs to be a straightforward model which was correct. From that, then arised three different models, that is semi-conservative, conservative and dispersive. If we talk about semi-conservative model, this is the model which it says that each progeny is going to retain one of the parental strand. That means one of your parental strand would be conserved when it has been passed from the first generation or second generation, and the next one will be the new one. So if the DNA molecule is untwisted, that is done by the helicase enzyme, two strands will be separated. One will be the template strand, and onto which you will be having a new strand which is a complementary DNA. Now, when this has been transferred, your parental strand is conserved in the first generation and so on. This is what is your semi-conservative model. Coming next, if we talk about other two DNA replication model which was proposed, that was conservative model. From the name it suggests that both the parental strand will remain together even after the first generation of replication. So this was the second model which was proposed where two new DNA, which is the two new progeny which has been formed of the DNA, will be together after the first generation of replication, and the parental strand will be remain together. That is what is the theory behind the model, that is conservative model. If you talk about the third model, which is a dispersive model, from the name it suggests that there will be some of the type of interspersed between the progeny DNA as well as parental DNA. Although two of the progeny DNA are identical over here, but there will be some of the parental and progeny DNA which will be interspersed. So there are three different models which was proposed to understand the replication in prokaryotes. That is semi-conservative, conservative and dispersive. Let's see diagrammatically how you can have the conservative model, semi-conservative model and dispersive model. Now students, if you can see on your screen, there are three different DNA replication model where the parental strand is represented in pink color and the new DNA is represented in blue color, as you can see on your screen. This is the diagram which we have taken from iGenetics. Now, the first model we can talk about, as I've already told you previously, that semi-conservative means half of it is conserved, that is half of the one is coming from parent and half is the new one. Now, if you can see here, parental DNA is pink color. After first cycle of replication, you can see that blue strand and pink strand are together. That means it says that pink strand, which is parental, and blue strand is a new progeny, which is going to tell us that this model is semi-conservative model, which is seen even after second replication cycle. Now, let's see the second proposed model, that is conservative model. From the name, it is suggesting that complete conservation of parental DNA. Now, if you see after first replication of the cycle, parental DNA, which is pink in color, is completely coming same together. That means two strands are remaining together without getting separated. This has been seen in the conservative model, and two new strands are being formed after the first replication cycle, which says that the new progeny is different and the parental strands are different. That is what is saying that 100% parental strands are coming together in the model called as conservative model, which is same observed after second replication cycle also. If you talk about dispersive model, although they both are coming differently, but some of the part of your parental and some of the part of your new progeny is interspersed over here. So this is again a third proposed model for DNA replication. Although there were three different models which was proposed for DNA replication, but among that, the best one or the one which has been accepted is semi-conservative model. Now, why semi-conservative model? To prove that you have the next thing, the experiment called as Messelson and Stahl experiment. Matthew Messelson and Frank Stahl in the year 1958, they showed an experiment evidence that whichever model is important for the replication in the prokaryotic cell, that is semi-conservative model. Let's see how did they got this particular evidence for the semi-conservative model replication correct. They grew E. coli because E. coli is having a life cycle between 20 minutes. So that was an easiest one to grow. So Messel and Stahl grow E. coli in a medium where they use nitrogen source, which is ammonium chloride. Over here, they have two different nitrogen. One was normal and second was heavy isotope, N14 and N15. Let's see now diagrammatically how we can explain that semi-conservative model was correct. Now, if you see E. coli, which has been grown in a medium containing N15, that is a part of ammonium chloride. Now, continuing to grow for the first generation, now this particular E. coli was replaced with a medium called as N14. During this period of time, they used a centrifugation, that is density gradient centrifugation, with the help of cesium chloride. During this, they found out certain photographs of DNA band, and they also find out the composition where according to the cesium chloride density gradient centrifugation, they could understood that how the bands are going to settle. If it is heavier, it is going to settle at bottom. If it is lighter, it will come at top. And if it is at intermediate, that is expected that if two bands are together, that is N15 and N14, that is going to be in the center. So during the first generation, if you can see over here, N15 containing medium, there was both the strand having N15 and N15, which is heavy DNA. So as you can see, if the heavier the DNA, it is going to settle during the density gradient centrifugation. Then they allowed this generation to grow after replication one cycle in N14 medium. Now, E. coli is utilizing ammonium chloride in N14 version. Now, if parental strand and the new strand or the new progeny are going to be working as a semi-conservative model, so 50% of your progeny comes from your parent. So over here, there is two different strand which was observed, that is N15 and N14, which is going to tell us that it's an intermediate density. How come intermediate density? If you see the pink one, which is the N15 heavier one, and the new one which has been formed, as we have previously seen in the diagram, that is the new generation, both of them are together. That means 50% is conserved. Again, in the second replication cycle, it remains same that the legacy, that is in the replication cycle 2, replication cycle 3, continuing the growth of E. coli in the presence of N14 and N15, they come to know that N15 and N14 are together, where it suggests that yes, in the intermediate density of cesium chloride density gradient centrifugation, we are proving that the correction of the semi-conservative model. That is how we proved about the semi-conservative model for DNA replication. As you can see in the diagram, we have E. coli grown in the medium of N14 and N15, heavy isotope and the normal one. In the density gradient centrifugation with the help of cesium chloride, we have given certain time for the replication depending upon the E. coli generation time. And then we got to know that the correct model over here is semi-conservative replication model. Now, we are knowing that the pattern is semi-conservative, but how does it does? Now, whenever there is a new DNA synthesized, we have to take up 4 important points. First of all, students, you require DNTPs, that is A, T, G and C. Without missing of this, the synthesis of DNA cannot occur. magnesium, which acts as a cofactor, along with a DNA fragment, which is a double-stranded, one will be acting as a template strand, onto which you will have a new complementary DNA forming. Other than that, DNA polymerase plays a significant role, which is also known as Kornberg enzyme. Now, this DNA polymerase, as you know, that is having proofreading activity, and as well as it is going to help us for the synthesizing a new DNA. Without DNTPs, without magnesium ion, without the DNA fragment or the polymerase, we cannot carry out the DNA synthesis. Now, let's see that how particularly we have a molecular detail of replication in prokaryotes. Before starting any replication, the randomness is not done at any point of the DNA. It requires a thing called as origin of replication. Origin means from where it is going to start. That is ORI. Over here, first of all, before starting your DNA replication, you require to denature or untwist the double strand DNA, because if the double strand DNA is not untwisted, how you are going to form a new DNA out of it? So for the initiation of DNA replication, you required both the DNA strand to be denatured at a specific location, which is called as origin of replication. During that time, it is going to form a bubble, which is called as replication bubble. Why it is important? Because that is the point where the enzymes are going to work. That is the point where other initiator proteins will come and start the process. So, first thing, we will do the untwisting. Before that, you need to find out the origin of replication. That is the time when the replication bubble forms. It will form a single strand DNA. Now you will have a question that how it is going to go from DS to SS. For that, you required one of the important enzyme, that is DNA helicase. Helicase role is for untwisting. Before that, there is an wrapping of your initiator proteins towards that DNA. So the DNA needs to be wrapped around it. It needs to be untwisted. Before that, your DNA is supercoiled. So topoisomerase is an enzyme which relaxes your DNA. Imagine you are having a phone wire, a PCO phone wire, which has been coiled so much around each other. You cannot take the call because it's so much coiled around each other. So first you need to relax it. You need to open the coiling and then you need to start the process. Imagine the same way. The DNA is longer. It is highly supercoiled. Without the supercoiling, without a relaxation of the supercoiled DNA, you cannot start it. So topoisomerase is the first enzyme, followed by initiator of the protein which wraps your DNA, and then we will uncatalyze that with the help of DNA helicase. Now, if you see the diagram of the model of formation of replication bubble over here, as I told you the first step that students, you required an origin of replication. Before that, you need to relax it because it's a supercoiled DNA. Followed by that, you require an initiator proteins which will sit at that location. Initiator proteins ka kaam kya hoga? It will initiate something. Now, DNA helicase, now he knows that, okay, I have to go at this location and start doing untwisting. Untwisting or making it from double-stranded to single-stranded. That is this yellow enzyme called as your DNA helicase. Now, helicase is loaded at this location. It will start opening the DNA where it will form as a bubble. Once the helicase role is actually going over there, the denaturation of helicase is taking place over there, then there is a person who is coming into the picture called as primase. Now, primase is the one which is very important, where you are going to start forming an RNA primer. Now, student, we need to focus that how you are going to form a complementary DNA on that. You have a template DNA, the direction of synthesis is 5' to 3', but on what you are going to form a new DNA. For that, this is an important structure. Initiator is holding that, because again it can come back to its original form. So initiator is holding that protein, followed by helicase is doing its work, but where you will add the DNTPs. So you required an initiator one, that is an RNA primer, which is over here. Once RNA primer is there, polymerase, which is in the purple color, will start adding DNTPs. And the direction of DNA synthesis is always 5' to 3', which is a leading strand. So here there will be an addition of a new strand, if you can see, which is in pink color, but still you have initiator protein at this side. We still have helicase which is moving forward over here, okay, and a RNA primer onto which next thing is formed. Now let's see one by one what are their role. So as I told you, topoisomerase followed by initiator followed by helicase. Now primase, which is encoded by the that is your gene called as DNA G gene, which binds to the helicase and the denatured DNA, which is going to be having an important complex structure. Why primase? Primase plays a significant role for activating the helicase. Along with that, it is going to create the synthesis of short RNA primer. This short RNA primer, which is around 5 to 10 nucleotide bases, which is going to help us to add the new nucleotide onto it. So this is how the DNA replication starts, or the initiation starts in your prokaryote. So again, we will put a glance on this. We have a double-stranded DNA, supercoiled. You need to relax it. The relaxation has been done by your topoisomerase. We have DNA gyrase, we have topoisomerase type 1, type 2, which is a two classes of topoisomerase, that is even topoisomerase and odd topoisomerase. Once it has been relaxed, initiator protein will find out a region of replication of origin. They will sit at that location, and then the helicase come into the picture and starts untwisting to it. Once it start untwisting to it, now there is a role of primase coming, where it will start initiating a small region where the replication will take place, because DNTPs needs to be added to form a new complementary base pair. And always remember, the direction of DNA synthesis is 5' to 3'. If we are going opposite of that direction, that means we are forming lagging strand that is been given a perspective by Okazaki fragment. So around Okazaki fragment and semi-discontinuous model, that we will cover in the next lecture. So students, today we have covered the topic. We will have a glance on the topic of today's concept, that what is DNA replication, what are the semi-conservative model, what are the three different models, how this semi-conservative DNA replication model got proved by Messelson and Stahl, followed by how we are going to do the molecular detail replication in E. coli, which is a prokaryotic DNA replication with the help of different enzymes like topoisomerase, DNA polymerase, helicase and primase together. So in the next lecture, we will cover the part of semi-discontinuity model. Until next time, thank you so much.